thank you very much to the organizers for the chance to be here. But also thank you to everyone who's spoken so far and participated. It's really been a marvelous week so far. I've learned a lot. One thing I'll say, though, is that giving a talk on such a general topic as disease ecology, where it's going, towards the end of such a great meeting means that a lot of the ideas I'm going to be talking about have been mentioned more or less directly as we go along. And I've been furiously taking note of this, but if I stop and mention each of those points of overlap, we'll be here for the full hour or beyond. So I'm going to mention a few, but the others are in mind, and I think they might form a great basis for further discussion this week and beyond. The other thing I want to say is that this is going to be a change of pace a little bit from the last few talks, because we were asked to focus on, current challenges, future challenges facing disease ecology. And so we actually called this, old problems and new solutions, but it might better be posed as old problems and new problems, because I'm mostly going to focus on things that we don't know how to do yet, problems that we don't know how to solve now. They may be unsolvable, but we'll see where we go. Okay, so disease ecology is a very broad field. And we were asked in particular to focus on multi-host, multi-pathogen systems, which are very complex and broad as well. And so my strategy in tackling this has been to sort of start with the simpler and then scale up to harder things and then quickly hand the ball to Pete. And so I'm going to start off by talking about models of zoonoses. Given their public health importance, they've been very well studied. They're, in principle, a simple multi-host system. So what can we learn from the literature on zoonoses, and what gaps does that literature still have open? And then I'm going to step sort of deeper into the disease ecology and talk about what we call reservoir dynamics, what goes on in the wild and other populations where zoonoses come from. And a real focus here will be, how do pathogens persist out there in a multi-host world? What do we understand about the dynamics of these problems? And at the end, I'll say just a few words about pathogen interactions in these multi-host systems. Clearly that's getting pretty complicated. Now throughout my talk there'll be two themes that keep popping up as well. One is what I'll call epidemiological dark matter, and the other is a set of arguments for why subcritical transmission and non-maintenance populations can't be ignored. Now I'd written all this before this week, but it's actually been gratifying to see how each of these issues, the challenges of missing data, and then a sort of back and forth discussion on should we care about subcritical systems, have come up quite a lot this week, so these ideas may be topical. Okay, so zoonoses. There's been a bit of talk about this paper so far. All it was with this merry band of collaborators, we sort of set off on the mad task of trying to read the whole literature on zoonotic models up until 2008 or so. So we read a ton of papers. These guys did an awful lot of work, and these guys contributed an awful lot of wisdom, perspective. And we learned quite a lot about sort of what's known, what's not known about how to model zoonoses well. And we've seen various versions of this figure, so I won't dwell on it. But bottom line is, zoonoses start in some kind of animals, they spill over to humans, they may or may not transmit further in humans. A key distinction then is, if they do have transmission in humans, is it subcritical or supercritical? So can it take off or not? We all know about that. Also, you can classify zoonotic pathogens as to how far they get through this process. And as was argued eloquently by James Wood and by John Drake yesterday, these stage 3 pathogens, who can have some degree of subcritical transmission among humans, are really an interesting group to look at. First of all, they've got good stochastic dynamics, things we like to play with, but they're also these are the pathogens who've sort of picked up the trick of spreading among humans. And just it could be the adaptation that Ollie's talking about. It could be a change of environmental circumstance. Something just needs to happen at a quantitative level to tip them over that threshold and possibly lead them to emerge more widely. So that's an interesting group. However, among the many patterns we picked out in this survey, the most striking perhaps was that nobody's working on those problems. So these graphs show, this is for directly transmitted zoonoses, the number of studies that have been published on each of the phases of the zoonotic process. Tons of work on epidemics in humans, tons on what's going on in the animals, very little at the interface between humans and animals, and those early stuttering chains of transmission among animals. This plot over here shows how many phases are included in each of the models. You can see almost none of them include more than one phase. So the whole idea of looking to zoonoses as a sort of laboratory, maybe we worked out all the problems of multi-host pathogens, doesn't really work out because they're broadly not being modeled as multi-host pathogens. But we've got lots to do on spillover and stuttering chains, so I'll dwell on that. Now I would argue that the key challenge here is to disentangle the contributions of zoonotic spillover and human-to-human transmission so that we can go on to do things like crucially estimate transmissibility among humans. We all know that that's the criticality, that's the threshold parameter which will enable us to assess the risk of emergence, and also to quantify the spillover process so that we can study it and ideally so we can prevent it. Now here's a cartoon of how that process works. You can imagine there's some reservoir species or spillover events, primary cases among humans who may or may not give rise to limited chains of transmission among the human population. So that's the problem in principle. One major challenge that we face is that we don't typically see this whole process. Instead, we don't see the transmission links generally. We also typically don't know the sources of infection for each case. We may, in fact, not see many of the cases. And for many diseases, we don't even know where it's coming from. And so, rather than having this lovely, well-resolved, beautiful picture we could do some straightforward inference on, we're working with this kind of data instead. And that really presents challenges when you try to match our, the already sort of noisy and heterogeneous dynamics of these processes against the data that are coming out of the field. Now there has been, that paper is in 2009, there's been some great progress since then, and there have been some new methods put forward, or improved methods, to infer parameters from the data that come from these kinds of processes. And I have to say, they're based on branching processes, and they're really building on results from a literature that was focused on diseases like measles in post-elimination populations, so another subcritical setting. And they've really fallen along two broad lines. One set of studies has looked at outbreak size distribution, so what can we learn from those? And then another really nice paper by Simon just this year just worked from the simple measure of what's the proportion of cases that were infected directly from the reservoir. And they each have their strengths and weaknesses. The outbreak size distributions, they allow estimation of R0 and possibly also transmission heterogeneity. And this works even if you don't know the source of the infections. But this process really, or this approach really struggles if the case detection rate is low, or if it's unknown, or if it's biased, if you're not getting an accurate picture of where these cases are coming from. The proportion of cases method also allows estimation of R0, and very intriguingly, sometimes estimation of the case detection rate itself. And it actually works quite well when the case detection rate is very low, but it requires that the infection source needs to be known. You also need to condition on the degree of heterogeneity. So I think a lot of progress has been made in these few papers, and then in John Drake's very nice talk yesterday, we saw a completely different approach to thinking about these subcritical processes. And I think the sort of complementary strengths and weaknesses of these two approaches suggest that there could be more progress at hand if we figure out ways to sort of put them together. But in thinking about broader problems in disease ecology, I want to emphasize the needs of these two methods. They both need, one way or another, pretty high resolution information about this subcritical transmission process in the target species. And in many systems, that's just not available. So in lots of systems, whether it's zoonoses that we're not paying as much attention to, or various systems involving wildlife or livestock, we may have spillover and subcritical transmission, but the infection sources and the case detection rates are not known, and it can be really difficult to estimate them. So our picture, we're really dealing with question marks throughout this process. And this brings me to this concept I want to raise of epidemiological dark matter. This is a phrase that I have happily pinched from this nice paper by Orton et al., which was actually talking about viral evolution at multiple scales. And they were talking about the impact of missed cases in transmission chains. But I think you'll all, given the discussion this week, you'll all agree that this idea can apply much more widely. So it can be some cases you don't see, but it can also be entire species you don't know about, host species. And Ollie's description just now of H7N9 and the various birds that may be upstream of the chickens speak to this point, that there are frequently host species we don't even know about. Or it may be processes that you don't know about. So you might have the right players, but you're not knowing how they're interacting. So I think broadly, we want to ask, what is the influence of these unobserved, unsampled entities in disease transmission systems? And what are we able to do to understand these better using modeling approaches? So a challenge I put out there, can we infer the presence of this dark matter from the data that we do have? So is there some way of picking up its footprint? And then secondarily, if we can see that it's there, can we predict what its impact will be, and can we correct for it somehow in our attempts to work with those data? And again, this week there have been a number of great talks on the inference side that have focused on missing data problems and the potential of new methods to deal with those. I've also appreciated Mark Woolhouse yesterday made the point of actually targeting your surveillance towards the very areas of uncertainty, which may resonate with this. And again, John's talk sort of brought forward a non-parametric approach, which in a way sidesteps some of the mechanisms we may not know about, if you can think of a signature of an outcome of interest that you may be able to detect from raw data. Okay, so I'll come back to this idea for sure. Now I want to step forward, or step backwards perhaps, to the reservoir. And when we talk about zoonotic models, we talk about reservoir dynamics. A better term for this might be all of disease ecology, because anything can happen upstream of humans in these systems. And so what do we know about how these processes work? And I think a first question, which seems simple but is actually not trivial, is what is this reservoir that we're all talking about? And here, I think the clearest answer put forward yet was in this nice paper by Dan Hayden and colleagues about 10 years ago, where they put forward a definition that they hoped would apply widely. So they proposed that a reservoir be defined as one or more epidemiologically connected populations or environments that captures the possibility of multiple hosts, even multiple non-animal reservoirs, in which the pathogen can be permanently maintained and from which the pathogen is transmitted to the defined target population. Now the part of this I want to focus on is this bit about permanently maintained. Of course, this is sort of a crucial element of all of our conception of what a reservoir ought to be, and it also makes contact with the classical problem in disease dynamics of what determines disease persistence over the long term in these populations. And so conceptually, this is, where am I? There I am. Conceptually, this is a very clear definition, but it turns out it's quite hard to implement it in a multi-host world. And so I'm going to sort of step you through what would be involved in taking their definition and how it actually plays out in a multi-host world. So the first step is you need to identify all the infected species. And again, this is simple in principle, but in practice, I would argue this is the general condition of disease ecologists working with generalist pathogens. We've got some circle of light we're looking within when we're looking for the reservoir host for some pathogen. Random, I've chosen a dromedary camel. And so we see that, and we may try to think about the dynamics involving that, but we never know, if we looked a little bit harder, if we might not find some other host that can also be infected by this pathogen. And it's very hard to know when you need to stop looking in this process, because there are always more host species out there. So that's actually a difficult challenge we have to deal with. The next question is to assign each of these species as either a maintenance host or a non-maintenance host. So we can put them in boxes or circles, as Hayden et al. Did. And the key question here is, can the pathogen be maintained indefinitely in that host species alone? And of course, this couples to the classical idea of critical community size. Does this host species in this location have a population above or below that mysterious threshold? That's also not an easy problem, as I'll come back to. The next challenge then is to estimate the cross-species transmission rates in this system. Another old challenge we've been thinking about for a long time and not easy. And finally, an idea that they put forward in that paper is to identify or detect maintenance communities. These are sets of host species, each of which individually is a non-maintenance species, but together they're well enough coupled that they can maintain the pathogen. So I'm going to move forward. I've already argued that this one's tough and we need to pay attention to it. But I'm going to spend the next few minutes on these next two steps. So the classical problem of identifying maintenance hosts in these systems, and then a bit on cross-species transmission. Okay, so identifying maintenance populations. We've been talking a bit this week about the critical community size. I think there's fairly broad agreement in the room that this is not a solved problem. even for the classic case study of measles. And I would argue it's a much harder problem even for wildlife populations. And we argued this point in a tree paper a few years ago. And that's for a range of reasons, some of which are fundamental to the concept. The first is that being a maintenance host is not the same as being simply supercritical. So estimating R0 is not enough. R0 greater than 1 is certainly necessary, but it's not sufficient for this point, and many people in the room have done important work on this problem. The next point is that any threshold, the population threshold for persistence, is not abrupt, and it's not even just a function of population size. And again, I know this is familiar to many of you. We need to think about the relative timescales of disease and demography in order to couple the population size to the probabilities of persistence. So this is just a simulation result showing the probability of persistence over some period for an SIR disease as a function of population size here and demographic turnover rate here. And the 2 points are quite evident. There are no sharp thresholds in these curves, and the impact of demographic turnover is at least as strong as the impact of population size. So we need to have all these things in our mind as we think about applying these ideas in wildlife populations. There are broader studies. I think there are a number of modeling and theory challenges here. The first would be a generalized way of thinking about the critical community size that accounts both for population size and demographic rates. And important steps were taken here by Ingemar Nazel in a 2005 paper, but I think there are further steps to be taken. Another, as we think about natural populations, is how can we account for things like population fluctuations, which can be very dramatic, and density-dependent host demography, which may sort of disrupt the assumed relationship between population size and replacement rates as we think about persistence in wildlife populations. And we can go further to say, well, what are the implications for those more complicated relationships for the control policies we use, which implement things like culling? A further set of challenges is to extend the theory to include mechanisms of the pathogen that include aid persistence, such as chronic carriage in some individuals, environmental reservoirs, and also heterogeneities of structure in the population. And of course, all this gets harder as you account for alternative hosts. Okay, so that's hard. How do we actually do it? One approach is you can reduce or eliminate the infection in certain host species and observe whether infection persists in other host species. Here's a great example from Brucella in Spain, where there are red deer and a number of livestock species all transmitting to one another. They knocked it out in the cattle, sheep, and goats. You can see these curves going down. That was sufficient to knock it out in the red deer. So based on that, they're pretty confident that red deer are not a maintenance population in this system. But what do we do in general? More often, we have good data on one host species and little or no information about other possible hosts. We're surrounded by this dark matter. So what we do is we study the natural history of the host-pathogen interaction we know about. We build a model, estimate the parameters, and then we sort of ask, can the model explain the observed patterns? So for instance, can the proposed reservoir species actually maintain the pathogen? That's a pretty good approach, but it's got issues. One challenge is the logic may be circular. If you fit the model to these data, then you would expect it to produce parameters that allow persistence. So you really need independent data to estimate your parameters. Another one is, what is the power of this approach to detect unobserved factors? So if something important is happening out there, can we see it? So sometimes you're lucky, and there's a good footprint of that dark matter. And this is a nice study by Megan Kraft about a canine distemper outbreak in lions in the Serengeti. And here, it was known there was some dark matter. There were some other hosts who got infected. There just weren't nice data. And this shows the spatiotemporal progression of the outbreak among lion prides where there were nice data. So what Megan did is she showed that you could not explain the data using just a model with lions in it. It simply didn't work. It couldn't pick up this jumpy spatiotemporal spread, couldn't produce the intensity and probability of outbreaks that you would need. And so instead, she put in hypothetical jackals or hyenas and showed the model could fit the data if you had these alternative hosts included. So the question I ask is, can we strengthen and formalize this kind of inference so that we can use it on other types of systems? Another kind of challenge is when the key processes are not so apparent in the data that we've got. An example here is a host-pathogen interaction where a small fraction of the hosts can become asymptomatic chronic carriers. and this is close to home. I study Leptospira in California sea lions. We are able to handle these animals fairly closely sometimes, so we've established that some of them become chronic carriers. But thinking about the dynamical consequences of this, we put a chronic compartment in an SEIR model, and we can see just by exploring it that adding this compartment has basically an undetectable effect on the dramatic dynamics of a localized outbreak. So it'd be very difficult to infer from the type of data that we often have, but it can have a huge effect, obviously, on long-term persistence. So again, can we devise methods to detect these kinds of cryptic processes, possibly by bringing in genetic or immunological data, some other dimensions? And then this week I was inspired to think, could we detect these using sequential Monte Carlo approaches? And I'm inspired here by Aaron's success in picking up signatures of inapparent cholera cases from time series data. So I'd love to hear thoughts on that. Okay, need to move on to cross-species transmission. This, again, is a difficult problem. One standard approach, or one way of thinking about it, is a bottom-up approach, where we say the force of infection across species is the product of prevalence in the source host, contact rate between hosts, and the probability of infection given contact. In wildlife systems, you sort of have a shot at measuring this one. There are approaches to measuring this one, as Ken described nicely this morning. This one is very hard for wildlife systems. It's hard to observe how many contacts don't lead to infection. So from the data we have, we can really only describe the opportunities for transmission, not the full process. So we're led to top-down approaches. For instance, we could think to study spatiotemporal correlations in the incidence in two species. And this works fine in principle, but it only really works for simple systems, not too many hosts, with good data and strong coupling. So it can work, and Mike Began's done very nice work on cowpox in voles and mice to infer this interspecies transmission rate. But in many instances, we just don't have sufficient data to do it. Now, a really exciting development came a couple years ago in this nice paper by Dan Stryker, where he was looking at rabies virus spreading among many species of bats in the US. So I don't know if you can see it, but there's a phylogeny here, and the colors indicate different clades of the rabies virus. These pies indicate essentially that of these clades, they tended to be concentrated in particular bat species you could think of as the maintenance host for those clades. And the little wedges show that they would spill over into other bat species. And from that, using some population genetics models, actually the migrate package that was just mentioned, Dan estimated this network of cross-species transmission rates, or actually called them reproductive numbers. So this is very exciting. It seems like exactly the kind of thing we want, and I think it is very promising. But there are a lot of open questions around these kinds of methods still. So the first is that the estimation of these transmission rates was based on some very strong assumptions. And I think we would all benefit from sort of looking at that more closely and maybe coming up with a more robust approach. There are also serious sampling issues. So one is the question of if you sample the different bat species unevenly, how will that bias your inference? But there's a dynamical bias also from subcritical transmission, where if the virus, a given strain, transmits a bit within a given species, that'll amplify the number of bats in that species who are infected after any spillover event. And it will, given that all the little spillover wedges are counted as a cross-species transmission event, that'll artificially inflate the estimate of cross-species transmission. So the subcritical transmission can't be ignored there. We also ask, how will this generalize for pathogens that evolve more slowly? So I want to come to this general point of why can't we ignore subcritical and non-maintenance hosts in these types of systems? Well, one, which sort of just came up, or some of the notions came up in hearing about H7N9 just now, is that they could potentially act as bridges to the target host. You may have a non-maintenance host that actually allows contact through to the humans for zoonosis, and we might call this zoonotic tunneling if we really have deep physics envy. Another reason is, as I just said, they may bias inference about cross-species transmission rates if we don't account for subcritical transmission. A third reason is they may contribute rare but influential events that actually alter the dynamics at higher levels. And here, one final example is bovine tuberculosis in New Zealand. So this is a system that Mick has done important work on, but this is a paper by Graham Nugent sort of summed up the multi-host ecology here. So possums are a capable maintenance host. Each of these other species generally are non-maintenance hosts, but they can contribute important rare events. So ferrets can disperse the pathogen to a different type of habitat. Wild pigs have long-range dispersal. red deer have long lifespans, and so they could sort of propagate it through time in great jumps. So we've got these rare events that could be contributing importantly to the phenomenon of persistence. And it brings to mind, given the strong analogy to things like dispersal kernels, or even the early work by Dennis on spatial contact models, whether kernel-based approaches might be appropriate to sort of study the broad phenomenology of this kind of system. Okay, and then the final reason, just as we worry about subcritical pathogens in humans because they might emerge, the same could happen among wildlife species. They may be just on the way to getting better in those hosts. So one more point I want to raise coming from the Stryker paper is that genetic tools may reveal sort of previously understood or previously unappreciated levels of complexity. This starts out blindly as a paper about rabies in various bats. So many host species, one pathogen. Turned out that there are many co-circulating strains of rabies that are co-circulating in a highly structured way. And I think if we look more closely, we'll see that genetics may show us similar levels of structure among host species as well, based on MHC types, HLA types, whatever. And so I think a problem we're going to have to deal with is what are the appropriate levels of aggregation for these kinds of systems? And are there important ecological interactions between types that we're not currently differentiating between? So here in this system, it seems quite likely that there's some degree of competition occurring between these different strains, given that there will be cross-immunity among them. And so, are we actually, as we look at these patterns, are we looking at the fundamental niche and the fundamental parameters describing these pathogens' abilities? Or is it more of a realized niche that is influenced by the competition among strains? And I think right now this falls in the dark matter category. We're not even seeing these things. Okay, with that, I want to have one slide on the next frontier, which is multi-host, multi-pathogen systems. Clearly, these are extremely challenging to think about. If I've just argued multi-host alone is a problem. A question I ask is it possible to tackle them and make more robust statements about them by studying either pathogen emergence or elimination events where the pathogen community has been perturbed? And an example that I've been working on, and which John mentioned yesterday, is the competitive release of monkeypox virus after the eradication of smallpox. So we've been looking at these data, and we see there's clear evidence that monkeypox is doing better as a consequence of the removal of smallpox. And it's still in the subcritical regime, I would argue, but it certainly is showing signs of danger. And from this kind of comparative analysis, we're able to see the impact that smallpox was having on the dynamics of monkeypox. And so we have to ask, given that this pathogen has popped up after this one's been eradicated, what are the implications of this kind of problem for other eradication campaigns? And there's similar dynamics possibly being observed for rinderpest and PPR virus, another morbillivirus. So it's a general issue. Okay, I want to stop there. I just want to leave these 3 points on the table as possible discussion starters, although I think we're going to go straight to Pete's talk. One is that abundant epidemiological dark matter, I think, is the normal state of affairs in disease ecology. We're always surrounded by possibilities that we're not able to measure. And I think we need methods to think more rigorously about that problem. Another is that spillover and subcritical transmission are important throughout these multi-host systems. And again, we need methods, especially for linking up with usually imperfect data on these problems. And at the bottom, I think we all agree that basic concepts of critical community size and these persistence thresholds need development, and especially so for natural host populations, which have more complicated population or even community ecology, and for non-acute pathogens. So we need theoretical work, we need pragmatic tools to actually determine what is a maintenance population, and more empirical study, which Pete may mention. And of course, there are further challenges here for multi-host systems. And with that, thank you. And I think Pete will step forward.
We'll do a combined questions at the end.
Okay.